Welcome, everyone. Today we are launching into, well, a real deep dive on what happens when the body's energy storage goes wrong. We're talking about metabolic disaster zones, specifically the glycogen storage diseases or glycogenoses.
That's right. And they're a really fascinating group of genetic disorders. Why? Because they perfectly show how just one tiny error, a mistake in a single enzyme's blueprint, can cause this huge systemic meltdown. It affects fuel, muscle, even the heart.
It really brings home how interconnected everything is.
Absolutely. And when we look at Mendelian disorders, those single gene conditions, GSDs, really stand out. They hit the body's main stored fuel reserve. Typically, they're autosomal recessive.
Meaning you need a faulty gene copy from both parents.
Exactly. You inherit one from mom, one from dad, and bam, the disease shows up, creating this immediate, often quite severe metabolic roadblock.
Okay, so let's start with the star player here, glycogen. We all kind of know it's stored glucose, but hearing you describe it, it sounds way more complicated than just sugar cubes stacked up. What is this stuff structurally?
Yeah, sugar cubes doesn't quite cut it. Glycogen isn't just, simple glucose waiting around. It's enormous, a huge, complex, highly branched polymer.
Okay.
Think of it less like a sugar cube and maybe more like a dense, massive cellular hedge maze, or maybe like a really, really condensed file storage system on your computer.
A hedge maze made of sugar. Okay, I'm trying to picture that.
Well, it gets big. A single molecule can have up to maybe 10,000 glucose units. Its molecular weight can hit 100 million Daltons. It's gigantic.
Yeah.
And that sheer size, plus all those branches, that's crucial. It lets the body pack a ton of energy away compactly, but also, and this is critical, it lets the body quickly snip off glucose units from dozens of branches all at once when you suddenly need that burst of energy.
Okay, so it's not just a fuel tank. It's like the body's emergency power grid, the buffer that keeps your blood sugar steady overnight or gives you that instant fuel when you, say, sprint for a bus.
Precisely. That's the buffering role. And GSDs happen when the enzymes, the little molecular machines responsible for either building this complex structure, that's synthesis, or for dismantling it, sequential degradation, when those enzymes fail.
And the consequence?
Well, the glycogen polymer just starts piling up inside the cells. It can't be used properly, can't be broken down, and this pileup causes, as you said, this internal traffic jam, specific to the organ, mind you, and that jam can seriously damage the cells.
Leading to all those different symptoms you mentioned earlier, low blood sugar, heart failure?
Exactly. Everything from life-threatening hypoglycemia to heart failure, depending on which enzyme is faulty and which organ is hit hardest.
That idea of a traffic jam is a really good visual. But why is the damage so varied? Low blood sugar, muscle cramps, heart problems. Those sound completely different, even if the root cause is glycogen processing.
That's a key point, the variability, or what we call pleiotropism. It all comes down to where that specific enzyme does its main job, its tissue specificity. The damage really depends on which organ relies most heavily on that particular glycogen pathway.
Okay, give me an example.
Right, so the liver. Its main job with glycogen is feeding glucose out to the entire body, keeping blood sugar stable. But skeletal muscle, it uses its glycogen stores strictly for its own immediate energy needs, like during exercise.
Got it.
So if a liver enzyme involved in releasing glucose fails, you get that systemic low blood sugar, hypoglycemia. But if a muscle enzyme fails, the problem is localized. You get muscle weakness, cramps during exercise intolerance.
Okay, that makes sense. So how do doctors sort through this? Because it sounds like you need a clear map to navigate these conditions. Same family name, GSD, but very different destinations.
That's exactly right. They classify them based on the functional pathology. What's the main metabolic failure and which organ system bears the brunt of it? Generally, we break them down into three main functional subgroups. It's helpful to keep these in mind.
Okay, lay them out for us.
First, you have the hepatic forms. Here, the big problem is mobilizing glucose from the liver. So the main symptom is severe low blood sugar hypoglycemia.
Failure to feed the system. Got it.
Second, the myopathic forms. These hit the skeletal muscles. The defect blocks the energy supply needed for muscle activity, leading to weakness, painful cramps, that sort of thing.
Exercise failure. Okay.
And third, there are the miscellaneous or generalized forms. The classic example here is Pompe disease, type 2. These affect pretty much all organs because the faulty enzyme is involved in basic cellular housekeeping everywhere. These tend to be the most severe, unfortunately.
All right, three categories: hepatic, myopathic, generalized. Before we unpack the specific diseases in each category, maybe we should quickly trace the normal path. Like you said, you can't really grasp the disaster without seeing the blueprint first. How is this huge glycogen molecule normally built and then taken apart?
Good idea. It helps to understand the normal flow. So glucose comes into the cell. First step, it gets a phosphate group added, becomes glucose 6-phosphate, or G6P. This molecule is really at a crossroads.
The crossroads. How so?
Well, G6P can either go straight into glycolysis, get burned for immediate energy, or it can be diverted towards storage as glycogen. If it's heading for storage, it first gets converted to glucose 1-phosphate, and then enzymes called glycogen synthetases and branching enzymes build up that huge branched polymer we talked about.
Okay, so that's the building part, the synthesis. Now, how about getting the fuel out of storage, the degradation? You mentioned two different ways this happens, which sounds important for understanding the different GSD types.
Crucial distinction, yes. Two main routes. The first, and the one we usually think about for energy, is the cytoplasmic route. This is the pathway the liver and muscles use for their specific energy demands.
And who are the key players here?
The star enzymes are the phosphorylases. There's a liver phosphorylase and a muscle phosphorylase, tissue-specific, remember. These enzymes act like molecular scissors, snipping off glucose units from the outer branches of the glycogen chain. They release glucose 1-phosphate.
Snip, snip, release the fuel. Sounds efficient.
That's very efficient. But there's a catch. They can't break down the entire structure. They work along the branches until they get close to a branching point, about 4 glucose units away. What's left is this structure called limit dextrin. The phosphorylases can't touch that.
Okay, so they hit a wall. That's where another enzyme has to step in to clean up.
Precisely. To dismantle that limit dextrin and get at the rest of the glucose stored deeper in the molecule, you need the debranching enzyme. It's got 2 jobs, really. It transfers a little chain of 3 glucose units, and then it snips off the single glucose unit left at the branch point itself. Then the phosphorylase can go back to work on the now linear chain.
Got it. A cooperative effort to fully unlock the stored energy. So that's the main rapid fuel release system. What about the second route you mentioned, the cleanup crew?
Right, the lysosomal route. This often gets overlooked in basic metabolism talks, but it's absolutely fundamental for understanding Pompe disease, type 2. See, small amounts of glycogen also get taken up into the cell's lysosomes.
The little recycling centers of the cell.
Exactly. Inside the lysosome, a different enzyme takes over. Acid alpha-glucosidase, sometimes called acid maltase, its job is just general cellular housekeeping, breaking down glycogen that gets cycled through the lysosome as part of normal turnover. A failure here isn't about immediate energy supply, it's a failure in cellular recycling, and it affects all cells with lysosomes, which is basically all of them.
Okay, that distinction feels really important. Cytoplasmic route for energy, lysosomal route for cleanup. Now we can tackle those 3 pathological subgroups properly. Let's start with subgroup 1, the hepatic forms. You called this the hypoglycemia crisis, failure to feed the system.
Yes. And the classic example, the prototype, is von Gierke disease, which is type I GSD. The core problem here is a deficiency in one of the liver enzymes needed for degradation. The liver cell is perfectly good at taking up glucose and building that big glycogen hedge maze.
It can score it just fine.
Stores it beautifully, maybe too well. The problem comes when you stop eating, maybe overnight fasting, and the body signals, okay, release some glucose into the blood. But the liver, it just can't. It can't break down its stored glycogen properly to release free glucose.
So the storage tank is full, overflowing even. But the tap to release the fuel into the bloodstream is jammed shut.
That's a perfect analogy. And the most critical enzyme involved in that final release step, the one that really opens the tap, is glucose 6-phosphatase, often called G6-pase.
G6-phase, okay.
This enzyme does the very last essential step. It strips the phosphate group off glucose 6-phosphate. Only then can the glucose molecule actually leave the liver cell and enter the bloodstream. If G6-pase is missing or doesn't work, G6P gets trapped inside the liver cell. It builds up and up.
And the liver just can't do its job of maintaining blood sugar levels for the rest of the body.
Exactly. Systemic hypoglycemia is the immediate result. And that buildup must do something physically to the liver itself, right? All that trapped material.
Oh, absolutely. Morphologically, the effects are dramatic. The liver gets huge hepatomegaly.
Yeah.
And often the kidneys, too, renomegaly. Why? Simply because the cells are physically stuffed, distended with glycogen. What does it look like under the microscope?
You see these massive accumulations of glycogen filling up the cytoplasm, the main body of the cell. But what's really striking and quite unusual for GSDs is that in type I, you can also see glycogen accumulating within the nucleus of the liver cells. It really highlights how severe the backup is.
Wow. Okay, let's talk clinical picture. It's clearly not just about low blood sugar, although that's dangerous enough. This is where that pleiotropism idea comes back, the ripple effects. How does failing to release glucose lead to things like gout or bleeding problems? Seems unrelated at first glance.
Right, it's where the metabolic detective work is really fascinating. Because that glucose 6-phosphate, the G6P molecule, it can't go forward to become free glucose, so it backs up. The cell has to do something with it. It gets shunted, forced into alternative metabolic pathways.
The escape routes.
Exactly, escape routes. And a sager one it gets pushed into is the hexose monophosphate shunt, or HMP shunt.
Okay, the HMP shunt. What happens when you ramp that pathway up?
Well, one of the key things the HMP shunt produces is ribose 5-phosphate. And that molecule is the essential building block for making purine nucleotides, the A's and G's in DNA and RNA, but also things like ATP.
Right.
So by forcing G6P down this path, you dramatically accelerate purine production and consequently purine breakdown. And what's the final end product of purine breakdown in humans?
Uric acid.
Uric acid. So these type I patients develop severe hyperuricemia, really high levels of uric acid in their blood. This can happen even in infancy or early childhood.
Leading to gout. In a child.
Yes, potentially leading to painful gouty arthritis and also kidney stones. It's all because of this secondary metabolic consequence, this overflow effect from the primary glucose block. It's a classic case of the body's attempt to compensate actually causing another disease.
Incredible. And what about the other issues like high fats in the blood?
That's another compensation gone wrong. The body senses it's starving for glucose, even though the liver is packed with it. So it triggers the release of free fatty acids from adipose tissue, from fat stores, trying to provide an alternative fuel. This leads to severe hyperlipidemia, very high levels of cholesterol and triglycerides circulating in the blood.
And the bleeding.
That seems to be related to platelet dysfunction, likely caused by the overall metabolic chaos. So you have this complex picture. Failure to thrive, stunted growth in untreated kids because of the chronic energy crisis, the dangerous hypoglycemia potentially causing convulsions, the hyperuricemia leading to gout, the hyperlipidemia, and a bleeding tendency. It's a cascade.
A systemic catastrophe, truly stemming from one stuck valve in the liver. Okay, if type I is the failure of the liver to fuel the system, let's pivot to subgroup 2, the myopathic forms.
Yeah.
The localized exercise failure. What's the core difference here in terms of the enzyme's job?
The core difference is that tissue specialization again. In these myopathic forms, the defective enzyme is one needed to generate ATP for physical activity, but specifically within skeletal muscle cells. The muscle can store glycogen, sometimes even more than usual, but it just can't tap into that fuel source properly during exercise.
So the problem isn't the body's overall blood sugar level.
Generally, no. Blood sugar is usually normal. The problem is a localized energy crisis. An energy bankruptcy right inside the muscle fiber when it starts working hard.
And the classic example here is McArdle disease, correct? Type V. Which enzyme is the culprit in this localized crisis?
That's right, type V. The missing enzyme is muscle phosphorylase. Remember we talked about the cytoplasmic breakdown pathway starting with phosphorylases? This is the muscle-specific version.
So liver phosphorylase is fine in these patients, but muscle phosphorylase is out.
Exactly. During strenuous exercise, muscles rely heavily, almost exclusively, on their internal glycogen stores for that quick burst of fuel. If muscle phosphorylase is absent or non-functional, the muscle simply cannot start breaking down its own glycogen. Glycolysis, the main energy pathway, can't even get going using glycogen as the source.
So the fuel is sitting right there.
Yeah.
You even see it under a microscope, right? Sometimes piling up under the muscle fiber membrane.
Yes, often you see accumulations in the subsarcolemmal space, just under the cell membrane. The fuel is there, but the muscle is effectively starving the second it needs that high-energy output. It's locked away.
And the clinical picture reflects that perfectly.
It really does. The onset is usually a bit later than type I, often in adulthood, maybe after age 20. The hallmark symptom is experiencing painful muscle cramps, stiffness, and really profound fatigue soon after starting strenuous exercise.
I've heard about a second wind phenomenon.
Yes, that's quite characteristic. If they stop exercising when the cramps hit, rest for a bit, they can often restart activity, usually at a lower intensity. That's probably because the muscle switches over to using glucose coming in from the bloodstream or fatty acids as fuel sources, bypassing the blocked glycogen pathway.
Okay, those painful cramps are literally the muscle hitting an energy wall. But you mentioned there's a key diagnostic test, something about lactate, that really nails the diagnosis.
Absolutely. It's a very telling metabolic signature. Normally, when you exercise intensely, your muscles switch to anaerobic metabolism, working without enough oxygen, and they produce lactic acid as a byproduct. This lactic acid spills into the blood.
So your lactate levels go up after hard exercise.
They should, yes. But in McArdle patients, because muscle phosphorylase is blocked right at the start, glycolysis from glycogen never gets going. The G6P needed for glycolysis isn't being produced from glycogen. Therefore, no lactic acid is generated from that pathway.
So if you test their venous blood after exercise, the lactate levels don't rise like they should.
Exactly. The failure to raise lactate levels in venous blood after ischemic form exercise, or just strenuous exercise in general, is the classic diagnostic clue. It proves the metabolic block is right there, upstream, preventing glycogen breakdown in the muscle.
That's the metabolic fingerprint. What about muscle breakdown?
That can happen too. The intense energy crisis can damage the muscle fibers, leading to myoglobinuria in about half of the cases. Myoglobin, the muscle's oxygen-carrying protein, leaks out and appears in the urine, sometimes making it look dark like tea or cola. This is called rhabdomyolysis, and severe episodes need medical attention.
Sounds dramatic. But what's the long-term outlook?
Despite the sometimes dramatic exercise-induced symptoms, the prognosis for McArdle disease is generally quite good. It's usually compatible with a normal lifespan. The key is management learning to recognize limits, avoiding prolonged, super strenuous activity that could trigger severe rhabdomyolysis, and listening to their bodies.
Okay, so we've covered systemic failure in the liver, type 1, and localized failure in the muscle, type V. Let's move to that third, very different category, the generalized forms. You highlighted the lysosomal catastrophe of Pompe disease type 2. Why is this one so distinct, forcing us to rethink GSDs?
Pompe is really in a class of its own in a way, because, as we touched on, it's both a glycogen storage disease and a lysosomal storage disease. The defect isn't in those cytoplasmic pathways for rapid energy release. It's in that lysosomal route of glycogen degradation we discussed earlier.
The cellular cleanup pathway.
Precisely. The missing or deficient enzyme is lysosomal acid alpha-glucosidase, or acid maltase.
So this isn't a fuel delivery problem like type I or V. It's a problem with recycling, with waste disposal that just happens to involve glycogen. And why does this defect affect basically all organs?
Because nearly every cell in the body uses lysosomes for that constant process of breaking down and recycling cellular components. Acid alpha-glucosidase is needed everywhere to clear out the small amounts of glycogen that normally get taken up by lysosomes. When it's deficient, glycogen just accumulates inside the lysosomes, and it happens in tissues all over the body: liver, muscle, brain, nerves, and critically, the heart.
Okay, so instead of glycogen floating freely in the cytoplasm like in type I or V, here it's trapped inside these ballooning lysosomes. What does that look like under the microscope? Must be quite different.
It's a very unique and frankly devastating picture pathologically. The cells in affected organs become swollen, filled with vacuoles. Those are the lysosomes, just stretched out. Microscopically, the cytoplasm often takes on this characteristic lacy or foamy appearance because it's just packed solid with these enormous glycogen-engorged lysosomes.
And all that physical bulk, while lysosomes literally crowding everything else out, must interfere with how the cell works.
Terribly so. It physically disrupts the normal cellular machinery, leading to profound organ dysfunction, cell damage, and eventually cell death.
Now, you said it's generalized, affecting all organs. But clinically, the picture, especially in the severe form, is dominated by one organ in particular.
Yes, the heart. In the classic severe infantile form of Pompe disease, cardiomegaly, massive enlargement of the heart, is the most prominent and life-limiting feature. The heart muscle cells, the myocardial fibers, become so choked and stiffened by these glycogen-filled lysosomes that they just can't contract effectively.
The pump fails.
The pump fails. This leads very rapidly to progressive heart failure, breathing difficulties, cardiorespiratory failure, and historically, death almost invariably occurred within the first year or two of life. It was a devastating diagnosis.
But similar to the other types, there's variability here too. It's not always that severe infantile presentation.
That's correct. Again, it comes down to the specific genetic mutation and how much, if any, residual enzyme activity is left. There are milder, later onset forms of Pompe disease, often called juvenile or adult onset forms.
And how do they present?
In these cases, while lysosomes are still affected everywhere, the symptoms might primarily manifest as a progressive skeletal muscle weakness, a chronic myopathy. It might even resemble McArdle disease initially, with muscle fatigue and weakness, but usually without the severe, rapid heart involvement seen in the infantile form. It really underscores the spectrum: same enzyme, different mutations, different levels of severity, and different clinical timelines.
Okay. Given how severe these diseases can be, especially those infantile forms of type 1 and type 2, let's shift gears to talk about treatment. Have these diagnoses changed? Have any moved from being almost uniformly fatal to something more manageable?
For type 2 Pompe disease, the answer is a huge, resounding yes. This is genuinely one of the great success stories in treating inherited metabolic disorders, and it goes back to that key distinction we made. Pompe is a lysosomal storage disease.
And that specific location, the lysosome, makes it treatable in a way the others aren't.
Exactly. It makes it amenable to enzyme replacement therapy, or ERT.
So the solution sounds almost conceptually simple. If the enzyme is missing, just put it back in, replace the missing part.
That's the core idea. And it works remarkably well for Pompe. The therapy involves manufacturing the missing enzyme recombinant human acid alpha-glucosidase in a lab. Then it's given to the patient through regular intravenous infusions.
But how does the infused enzyme get from the bloodstream into the lysosomes inside the cells where the glycogen buildup is actually happening?
That's the clever part. The engineered enzyme is designed with specific molecular tags, typically mannose-6-phosphate residues. These tags act like address labels. Cells throughout the body have receptors on their surface, mannose-6-phosphate receptors that recognize these tags.
Like a lock and key.
Kind of. The enzyme binds to the receptor, and the cell actively pulls it inside through a process called endocytosis. Once inside, the cell packages it up and transports it directly to the lysosome, the exact place it needs to be to start breaking down that accumulated glycogen.
Wow, it's like a targeted delivery system using the cell's own mail service. What's the real-world impact been for Pompe patients, especially those infants who used to have such a grim outlook?
The impact has been nothing short of transformational. Before ERT, infantile Pompe was rapidly fatal. Now, with early diagnosis, often through newborn screening and starting these regular infusions, often weekly or biweekly, each can stabilize and often improve heart function. It helps clear the glycogen out of the lysosomes in various tissues, improves muscle strength and motor function, and dramatically extends survival.
So it's not quite a cure, but
Not a cure, no, because the underlying genetic defect remains, but it has fundamentally changed the natural history of the disease. It shifted infantile Pompe from an acute, fatal condition to a chronic, manageable one. Many children receiving RT are now surviving well into childhood and adolescence, reaching milestones that were previously unthinkable. It's a huge step forward.
that is genuinely fantastic news for Pompe disease. But what about the others, like type I von Gierk or type V McArdle? You said ERT doesn't work there because the problem enzyme is in the cytoplasm, not the lysosome. Are there other strategies being explored for them, maybe borrowing ideas from other genetic disorders?
Yes, researchers are definitely looking at alternative approaches, often adapting concepts used for other types of storage diseases. One important conceptual strategy is substrate reduction therapy, or SRT.
Substrate reduction. Okay, break that down. How does reducing the substrate, the starting material, help if the problem is breaking down the end product, glycogen?
Think of it like managing a blocked drain. If you can't clear the blockage effectively, the enzyme defect, another way to prevent overflow is to simply turn down the tap, reduce the amount of water substrate flowing in.
Ah, okay. Slow down the production line.
Exactly. In the context of GSDs, SRT would aim to inhibit the synthesis of glycogen in the first place. If the body can't properly degrade glycogen, maybe reducing the amount of glycogen being made could lessen the accumulation, reduce the cellular distension, and alleviate the symptoms. You're lowering the overall burden on the faulty degradation pathway.
Does this connect to how type I is managed now, with that continuous feeding?
In a way, yes. The careful nutritional management for type I, using things like uncooked cornstarch to provide a slow, steady release of glucose, aims to prevent hypoglycemia. But it also minimizes the body's need to synthesize and then try to break down large amounts of liver glycogen during fasting periods. So it acts as a sort of indirect substrate management. Future SRT drugs, though, might directly target enzymes involved in glycogen synthesis, like glycogen synthetase, to more specifically reduce the glycogen pool. It's about rebalancing that input versus the blocked output.
Interesting. Okay, what about trying to fix the faulty enzyme itself? If there's some enzyme being made, but it's just unstable or misfolded. I think I've heard of molecular chaperone therapy.
That's another really exciting area of research, yes. Molecular chaperone therapy. This is particularly relevant for genetic disorders, including potentially some later onset or milder forms of GSDs, where the mutation doesn't completely abolish the enzyme, but instead causes the protein to misfold as it's being made.
So the enzyme molecule is produced, but it's misshapen.
Right. And the cell has quality control systems, primarily in the endoplasmic reticulum, that recognize these misfolded proteins. They say, whoa, this isn't right, and target them for destruction before they can even get to where they're supposed to work.
So the chaperone steps in to sort of hold the faulty protein together.
That's the idea. These chaperones are often small molecules, sometimes taken as oral drugs. They bind specifically to the unstable mutant enzyme protein. This binding helps stabilize the protein's structure, acting like a scaffold or a folding template.
Preventing it from being immediately destroyed.
Exactly. It helps protect it from that rapid degradation by the quality control system. This allows more of the somewhat faulty but now stabilized enzyme to escape the endoplasmic reticulum, travel to its correct cellular location, like the cytoplasm or lysosome, and carry out some of its intended function. Even a small increase in residual enzyme activity can sometimes make a big clinical difference.
Is this being used for GSDs now?
It's not yet a standard therapy for the classic severe forms like type I or type V, but it's a very active area of research, especially for diseases with known misfolding mutations. And it holds promise for certain GSD variants where rescuing even partial enzyme function could be beneficial. It highlights the push towards personalized medicine based on the specific type of mutation.
Okay, so we have ERT for Pompe, potentially SRT or chaperones for others. But all these GSDs fundamentally boil down to a single mistake in the genetic code. Where does that point us for a true long-term fix? A cure?
That really points towards gene therapy, given that GSDs are classic examples of monogenic diseases caused by defects in a single gene. With often severe consequences, they are considered prime candidates for gene replacement strategies.
Replacing the faulty gene with a working copy.
That's the ultimate goal. The idea is to deliver a normal functional copy of the defective gene, like the glucose 6-phosphatase gene for type I, or the muscle phosphorylase gene for type V, directly into the patient's cells, especially the cells in the most affected organ.
So for type I, you target the liver cells.
Precisely. You'd aim to get the correct gene into the hepatocytes. Typically, this is done using specially engineered, safe viral vectors like adeno-associated viruses, AAVs that act as delivery vehicles. These vectors carry the therapeutic gene into the target cells.
And if it works?
If it works, the patient's own cells, like their liver cells, would start using this new blueprint to manufacture the correct functional enzyme themselves. This could potentially correct the underlying metabolic defect permanently, or at least for a very long time, offering a stable, potentially curative solution that might eliminate the need for constant dietary management, ERT infusions, or other lifelong therapies.
Where are we with gene therapy for GSDs? Is it happening?
It's definitely moving forward. There are active clinical trials underway for gene therapy for several GSDs, including type I. The initial results are often promising, but there are still challenges to overcome regarding long-term efficacy, safety, and ensuring the gene gets delivered to enough cells in the right tissues. But it represents a huge area of hope and ongoing research. It could be revolutionary.
This whole deep dive really drives home just how intricate cellular metabolism is. It's still kind of mind-blowing that one faulty enzyme, one tiny molecular machine breaking down, can cause such incredibly diverse and sometimes catastrophic problems.
It really does, and it perfectly illustrates a couple of fundamental concepts in genetics and pathology that are worth highlighting again. First, that idea of pleiotropism, one gene defect, causing a whole constellation of seemingly unrelated effects, like in type I, the hypoglycemia, the high uric acid, the high lipids, the growth failure, all traced back to that single G6-pase enzyme deficiency.
One root cause, many branches of problems.
Exactly. And second, it shows genetic heterogeneity. Within the umbrella term GSD, you have multiple different diseases caused by defects in different genes, like G6-pase versus muscle phosphorylase versus acid maltase. And even within a single type, like Pompe, different mutations in the same gene can lead to vastly different clinical severities, from fetal infantile forms to milder adult onset. Same core problem, glycogen accumulation, but reached through different genetic paths.
Okay, let's try to synthesize this one last time. Let's quickly revisit those three core patterns, just to make sure everyone listening has the key differences locked in.
Good plan. Let's recap the prototypes. First, type I, von Gierke disease. Where's the failure?
The liver, the enzyme, glucose 6-phosphatase.
Right. And the main consequence?
Failure to feed glucose into the blood. So severe hypoglycemia and all that secondary metabolic chaos.
Perfect. Next, type V, McArdle disease. Where's the primary failure?
In the muscle. The enzyme is muscle phosphorylase.
And the consequence?
Failure to fuel the muscle itself during exercise. leading to painful cramps, fatigue, and that lack of lactate rise.
Excellent. And finally, type 2, POMP disease. Where is the failure here?
This one is generalized. It's a lysosomal problem, missing acid alpha-glucosidase.
And the main consequence, especially in infants?
Failure of cellular housekeeping everywhere. But the most devastating impact is usually massive heart enlargement, cardiomegaly, leading to heart failure. Though now, thankfully, often manageable with ERT. Brilliant. Understanding those three core failures, systemic fuel supply, localized muscle fuel, and generalized cellular maintenance, that really is the key to making sense of the glycogen storage diseases.
Absolutely. And maybe we can leave you, the listener, with one final thought to chew on. We talked quite a bit about how the body compartmentalizes glycogen use. The liver stores it to maintain blood glucose for the whole body. The muscle stores it strictly for its own use during contraction.
Two different jobs, two different locations.
Right. So think about this. What evolutionary pressures might have made it necessary, or at least advantageous, for the body to evolve separate, highly specific enzymes like liver phosphorylase versus muscle phosphorylase to handle what seems like the same basic chemical reaction, breaking down glycogen? Why not just have one universal glycogen breakdown system?
That's a great question. Maybe having one system would be too slow or impossible to regulate precisely enough for both needs simultaneously. Like you wouldn't want your muscles dumping glucose into the blood when only the brain needs it, or the liver holding back fuel when the muscles are sprinting.
Exactly. The complexity suggests that fine-tuned, independent regulation was absolutely critical. The need for the liver to respond to hormonal signals for slow, steady glucose release during fasting is totally different from the muscle's need for an almost instantaneous, massive mobilization of its internal fuel reserves triggered by exercise. Having distinct, tissue-specific enzyme systems allows for that exquisite, independent control. The GSDs, in a way, are the tragic proof of just how vital that precision is. When even one specialized cog breaks in that incredibly intricate metabolic machinery, the consequences can be profound.